I'm Daniel Levine, and this is the Bio Report. Regardless of the cause of a disease, most diseases involve gene dysregulation. Omega Therapeutics is developing a new class of programmable epigenomic mRNA medicines designed to make specific epigenetic changes and correct abnormal gene expression to treat or cure diseases. We spoke to Mahesh Karande, President and CEO of Omega Therapeutics, about its pipeline of mRNA therapies, how they work, and its recently announced collaboration with Novo Nordisk to develop an epigenomic controller to treat obesity. Mahesh, thanks for joining us.
Danny, thank you for having me.
We're going to talk about epigenetics, Omega Therapeutics, and the use of mRNA to target the epigenome to regulate the activity of genes. Perhaps we should begin with the epigenome. Can you explain for listeners what this is and how it regulates gene activity?
Yeah, no, definitely. I think all exciting topics. So look, people are familiar with the genome and genes, right? That's a very well-known term, phraseology, all of that, right? So the epigenome, if you think is, just from the Greek epigenome, it's around the genome or one step above the genome, right? And that is the control system that nature uses to control the genome, to put it very simply. So essentially, epigenome is what really dictates how genes express, what happens with our cells, why a particular cell becomes a heart cell versus a lung cell, why a butterfly, from a caterpillar stage develops and becomes a butterfly. So how does that happen? Mother Nature has a way of making all of that happen. And that is essentially the work of the epigenome. So what nature does is it lays something called epigenetic marks at different points in the genome. And those epigenomic marks actually regulate gene activity. So essentially, think of it, to put it very simply, think of it as a thermostat or a rheostat. And if you want, to change the temperature, what you do is you go and dial up or dial down, right? That is precisely what Mother Nature does by using the epigenome, to put it very simply, that it literally lays these epigenetic marks that dial up or dial down a gene's expression, and life goes on.
Many people think of the role genes play in disease in the context of genetically defined diseases, but what happens to genes when people become sick?
Yeah, look, I think genetically defined diseases are sort of the rare diseases that most of us know when we think about genes and their correlation to disease, right? That, in fact, forms a very, very small subset of overall diseases and conditions known to us, right? But if you really think of disease in general, disease occurs because genes get either overexpressed or underexpressed. In vast majority of cases, right? Think of it as a software program misfiring. And if the software program misfires, then you're not going to get your end result, right? That's really what happens in our body. As our genes express, right? It undergoes a process, which is known very well in biology, called the central dogma of biology. So genes express through this epigenetic activity that I talked about, right? Then a process happens, which is called transcription, and mRNA is created. And then that undergoes translation, and protein is created. And protein, as we know, is the building block of life, right? So essentially, when disease occurs, irrespective of etiology, means irrespective of the origin of the disease, in vast majority of diseases, genes either overexpress or underexpress. So they're not in the normal range of expression. That's really when people become sick or we get some sort of a condition. So if there was a way to bring genes back into that normal range, right? Then by induction, you would resolve disease because you would fix this problem right where it starts. That's really the role of genes in diseases.
What's the potential to exploit that as a means of treating diseases that haven't been amenable to more conventional approaches?
Yeah, so once you have figured this out, right, then you kind of know how Mother Nature operates in this case, right? In this case, what Mother does is she lays epigenetic marks at all of these different locations in the genome, because of which genes express correctly. Now, nature goes wrong sometimes. Its programs go wrong sometimes, and if you have a way of really co-opting that control system, and if you figure a way out of doing the exact same thing that Mother Nature does, but are able to write corrective programs, right, then you would actually be able to do what nature does and bring the genes back into the right range of expression. That is actually the potential for this science. So this science really delineates how our genes are organized and how they are controlled epigenetically, so as to express correctly and life to go on. What we have figured out, and what once you figure out this science that we talked about, right, you could harness that science, create medicines, create a platform that creates medicines that very systematically goes after genes, single or multiple genes implicated in the disease, and bring them back to correct expression simply by laying epigenetic marks. That is really the potential of this. And what happens is, some of these genes, some of these genes that have proteins, they are not amenable to conventional approaches. So let me give you an example, right? In oncology, for example, cancer, which is a poster child for epigenetic dysregulation. There are many of these pan-essential oncogenes. Think of them as master regulatory oncology genes. KRAS is an example, c-MYC is an example. These genes, it's very, very difficult to do a few things. One is, some of them, it's very difficult to target them at the protein level with conventional medicines like small molecules or large molecules, simply because their structure, the protein structure, and its half-life sometimes does not allow drugs to bind to these proteins, right? So it's very difficult to tackle it at the protein level. When you try to tackle it, one step up in the central dogma at the mRNA level, what happens is, these genes are so tightly regulated that if their mRNA comes under attack, or even if their protein comes under attack, they regulate. Because these conventional approaches, the way they work is, they have to work non-specifically everywhere. So think of a drug, a headache medicine that you take, right? It does not only resolve your headache, but if you had a pain in your foot, it would resolve that as well. The reason that happens is because it goes everywhere in your body, and wherever there are those pain receptors, it acts on, and it dampens your pain, right? That's pretty much how conventional drugs work. So with these kinds of oncogenes, for example, if you approach it conventionally, you kind of have to shut down its expression everywhere, because of which they auto-regulate and crank out more and more mRNA and protein, right? And if that happens, you need to put more drug in the system. And at some point, you can't put enough drug in the system to control these genes. So the only way to control these genes and their expression is to go, very high up where they sit pre-transcriptionally, and sort of, instead of completely shutting them down, tune them down such that you resolve disease, but you prevent autoregulation. So this is just one example. Examples like regenerative medicine. If you could control master genes, look, as we grow from an embryonic stage to a full-grown stage, what happens is, genes get turned on for long periods of time, then they get shut off for long periods of time. Once, let's say, your liver is fully developed, I don't necessarily think the gene that turns on liver function has to be expressed all the time. I'm just simplifying this, right? Obviously, science is far more complicated than that, but just take this as a simple example. But what happens if, for example, your liver stops functioning well because, I don't know, whether you had a viral insult, some sort of hepatitis, or somebody drank too much and developed liver cirrhosis, and the liver stops functioning? Is there a way then to go after a gene that controls liver function, turn it on or tune it up so that some part of the liver starts functioning again. If you could do that, that would be powerful, right? Conventional approaches don't allow you to do that. That's where what we are doing, which is controlled epigenomic modulation pre-transcriptionally, becomes a very, very powerful approach to actually go after these diseases. There are many, many examples like this, and actually one of the examples that I hope we talk about is this latest partnership that we did with Novo Nordisk in the field of obesity.
I want to talk about what Omega is doing, but before we do that, I thought it'd be useful to explain one concept. Can you explain what insulated genomic domains are and the role they play?
No, absolutely. So remember, I was talking about nature's control system of genes, right? Essentially, what nature has done is that it has organized all our genes and their regulatory elements, the regulators that control gene expression, in these three-dimensional conserved, loops or structures of DNA that are called insulated genomic domains. Okay, individually, an insulated genomic domain can contain one or multiple genes, on an average about 3 genes, right? And there are about 15,000 of these IGDs that are distributed across the 23 chromosomes and are ubiquitous in every cell of our body. So if you think of every human cell, it has the exact same DNA, right? Which makes sense. But epigenetically, a heart cell becomes a heart cell and a lung cell becomes a lung cell as we develop. So that's sort of the basis of the organization of our genome, right? Now, these IGDs, the whole system is actually very, very interesting because it is evolutionarily conserved across mammalian species. In the lower order mammals, you might have genomic sequences that are different, but the conservation of this system between non-human primates and humans is almost intact, right? And this is a testament to evolution. Now, what happens here is, there are a couple of things that are very, very interesting about these IGDs. First, an IGD is a wholly, contained control unit for the genes that sit in it, because only the regulators that sit inside the IGD can control the genes inside the IGD. So imagine that's a whole unit of gene control, right? Now, if you could tap into that, you could control gene expression. So how do you tap into that? There's another very interesting thing that we figured out as we were delineating this biology. And that was that the sequences, the genomic sequences of each of the regulatory element inside an IGD is unique in the entire genome. So if you pick up a sequence in one IGD out of the 15,000 and look at its genomic sequence, it is unique to that sequence in that IGD and is not repeated anywhere else across the genome, right? That means in each cell, you have that same sequence repeated for that particular element. That means you can target that regulatory element very specifically and use that to lay an epigenetic mark and control gene expression, right? That is pretty much what we have figured out. And that's why this IGD biology is so important, because it gives you an ability to control genes, and it gives you these very specific and unique targets that we call epigenomic zip codes. They're like epigenomic zip codes. They're unique. So we coined this phrase epizips. That's really the basis of the biology by which we can control gene expression epigenetically.
You're developing a class of medicines that you call epigenetic controllers. How do these epigenetic controllers work?
So we call them epigenomic controllers exactly because of their function, right? They control the epigenome, right? So the way they work is, remember, I was just telling you about these epizips and regulatory sequences, right? So if you could figure out a way to home into an epizip or multiple epizips and lay epigenetic marks there, then you could modulate gene activity for the single or multiple genes that sit inside that IGD. So we figured that out, and what we have done is we have created a completely new class, right? Like you said, epigenomic controllers, and they are mRNA therapeutics. So everybody, has heard of mRNA, obviously, in the context of vaccines. So we use mRNA very differently than how vaccines use mRNA. We use mRNA to enter the body, a particular tissue and a cell through what is called the ribosomal machinery, right? That allows mRNA to enter and go home into the nucleus of a cell. And there it expresses two sets of proteins. So this mRNA is encoded for two sets of protein. One is called a DNA binding domain, which is a protein that goes into that is designed to home into that particular sequence of that episode, right? Like a homing pigeon. It goes in and it homes into that sequence, that single sequence, and locks itself over there. There it expresses a second protein. mRNA expresses a second protein called an epigenomic effector. Now these are naturally occurring, proteins that exist in our bodies that we are able to use, or we are able to slightly modify for them to exactly carry out the function we want them to, right? So once that epigenomic effector is expressed there, its job is to regulate the gene. So if the gene is turned up or down, it actually brings it back into a normal range of expression by either dialing it up or dialing it down for a duration that we choose in terms of designing this molecule. That's really what we are doing. So this is how epigenomic controllers work. So to step back, this is the first systematic use of mRNA therapeutics as programmable epigenomic medicines. And the words are very important, right? They're epigenomic medicines because they work on the epigenome. They do epigenetic work. And they're programmable because we are able to prospectively design these for the DNA with the DNA binding domain to go into the exact location that we want, and an epigenomic effector that regulates the gene exactly to the level we want, either up or down, and for the duration that we want. So that gives us three properties. One is high specificity, right? Going into one location in the entire genome. And then two more things, which is the up or down modulation, so controlled modulation, and controlled durability, the length of time for which the drug has to act. So for example, in chronic conditions, we could design epigenomic controllers that act for a long period of time. So people don't have to take a drug every day. They could take it probably once every month, or once every 2 months, or once every 3 months. And for something like oncology, where physicians usually treat patients every 2 weeks with an infusion, we have created our first drug, which is in the clinic, to act every 2 weeks, so that fits into the oncology treatment paradigm. So that's how programmable these are. That's how engineered these are. And we've designed these very prospectively to have the exact activity that we want them to have.
Are you able to target anywhere in the genome with these?
Yes, we can. Because remember, the genome has all of these regulators. We know all of them. So we have created a proprietary list of targets, epizyps, that only Omega has. So once we know which gene is correlated to which disease, and we know which IGD it sits in, then we have the entire real estate of that IGD, all of the regulators, right? They can be enhancers, they can be promoters, they can be, repressors that control that gene expression. And we can figure out which of those we want to target with what kind of epigenetic effect. And then we have our targets that we design these epigenomic controllers for, that go and actually do exactly what they're supposed to do. So we can pretty much target any gene in the entire genome with this approach. And the beauty of this is that makes our approach and our platform exceptionally broad, because this can be used pretty much for any human gene, for any disease process, any disease.
How well understood are the genes you need to target and modulate to treat any given disease?
Yeah, that's a great question, Danny. Look, fortunately for us, we are living in times where our understanding of disease biology is very, very good, right? I mean, I think let me put it this way: if Omega had to just focus on known biology, where genes are correlated with disease, we would be building a phenomenal company for the next 15 years. We would not even have to worry about unknown biology, although our approach allows us to even examine the entire genome by disease to figure out which genes are implicated. We don't even need to do that. So I think there's a very, very wide range of diseases that we already know the biology for, and we already know what genes get implicated, either single or multiple genes. They can sit. Now, there are some diseases that are complex, right? Multigenic as well as complex diseases, which it's not a single gene that controls, or a couple of genes. They could have multiple genes, and you could take multivariate approach in controlling them, right? That's the future.
These are packaged in lipid nanoparticles. Is there anything packaged along with the mRNA that makes up a therapy? Are there things like promoters or things to target specific cell type? What does a therapy consist of?
Yeah, so you're right. I mean, I think, look, from an Omega therapeutic standpoint, as such, we are delivery agnostic. So when you talk about lipid nanoparticles, you're talking about delivery to particular tissues and cells, right? So we are agnostic in the sense we could use any sort of delivery particles. But what we have done is, in our initial programs, we have gone with lipid nanoparticles simply because mRNA, that is inside of a lipid nanoparticle, is a modality, thanks to the vaccines, that is in more human beings today than potentially all other biotech modalities combined, right? So I think from that standpoint, it's de-risked, it's safe. People understand what any, if there is any toxicity profile of these drugs, what it will be like, right? So that's why we went in with mRNA and lipid nanoparticles. Now, the way this works is, to put it simply, obviously, mRNA is enclosed inside the lipid nanoparticles, and given the size of our constructs, we can actually fit multiple mRNA constructs in a lipid nanoparticle. That's why we can multiplex to different locations in the genome with a single approach, single therapeutic, right? And these lipid nanoparticles then are, they are tissue specific. So, for example, right, the liver has been targeted with lipid nanoparticles for a long time, right? And you have new generational LNPs, lipid nanoparticles, that we are using now in our program. We also have another program where we are working towards an IND targeting the lung. We have our internal development in lung, whether it's systemic, like you would use in oncology, or inhaled, like you would use in respiratory diseases. We are also targeting aspects of other tissues, right? For example, we have intrathecal delivery. We are looking at the skin. We are looking at direct joints. The Novo program that we are working on, we're going to be working on together to target adipose tissue. So lipid nanoparticles have come a long way. The entire industry is sort of working towards targeting all different types of tissues and cells, and Omega is at the forefront of it. But again, we are delivery agnostic. We could also use viral delivery, for example, in places which are privileged compartments. So, for example, the eye, etc., where risk of immunogenicity is very low. So there's a tremendous amount of breadth of delivery technology that we can leverage here with our epigenomic controllers, which are essentially these mRNA constructs.
You're using an AI-based computational engine as part of your drug discovery. What does this do, and how does this impact your ability to move experimental therapies into the clinic?
Yeah, I mean, absolutely. I think Omega Therapeutics is in a big way leveraging AI and machine learning. I mean, if you just step back and imagine the data that we are dealing with here, right? The quantum of data that we are dealing with, just genomic data. Then you have disease data. Then you have data correlating genes to diseases, all sorts of databases. And for us to rapidly crunch through all of those data, right, first of all, requires a tremendous amount of algorithmic and computational ability, which we have, right? Secondly, once we decide, now I'm simplifying, once we decide which target sequence to go after, right, we have now to design our epigenomic controllers, and particularly the proteins that the mRNA will express, right, that has to target that specific locus. And then we have to look at the epigenomic effectors that we will tether to that particular DNA binding domain. All of that is done algorithmically by using our computational genomics and AI capabilities, right? So we have a super solid AI and com genomics engine that is the core of Omega's platform technology. And what this does in terms of our ability, this is incredible. Now, what I'm going to say is just incredible, which is, think of it this way. Once you know which disease you want to go after and you know which gene you want to go after, now you want to interrogate the entire genome and figure out the IGD where that gene sits. And then you want to interrogate that entire IGD, look at all the sequences of all the regulatory elements, which are the epizips in that IGD, right? And figure out very quickly which epizips you want to act and what kind of epigenetic effect you want to have, right? All of this, if as we use computational genomics and AI, takes us between 3 and 4 weeks. So imagine crunching through all of these data within 3 to 4 weeks. We already know where we want to act and what kind of mechanism we want to use. Then the next 3 to 4 weeks, we use our algorithms and actually design these constructs, right, and make them. So in that period, so give or take, 2 to 4 months on the outside, 4 months, right? You have 5 to 10 epigenomic controllers ready for testing, and these are literally designed prospectively, and we start testing them in vitro and in vivo, trying to figure out whether now experimentally whether they are actually going to have the exact same effect that we designed them for. That process takes us about 6 to 9 months, and then we are able to declare a development candidate. So just imagine the timelines here, right? 4 months, another 6 months, or 9 months, so 10 months to 15 months, you can go from scratch to actually declaring a development candidate. Now, if you have a lipid nanoparticle ready, like we had in the liver program, then you start your IND enabling work, and that took us another, I would say 12 months. So for our first program that is in the clinic, and we are actually getting clinical data, patient data now, from scratch to getting an IND cleared, took us 27 months. I don't think there is anybody else who can do it this fast, right? And this is not fast for the sake of doing it fast. This is because we have the power of AI-based computational genomics and the engine of the Omega platform, and we are able to prospectively design and engineer in the right properties that we are then eventually testing in the clinic. So we know how this drug is going to work, we know where it's going to act, and we know exactly what it's going to do, and that's what then we start testing eventually, and then obviously, hopefully patients benefit from it. That's the power of this. That's what essentially allows us to move very, very fast.
When you get a platform like this that has such broad potential, it's always interesting to me the decision-making process on how you prioritize which indications you'll pursue. I'm wondering if you could talk a little about that. How did you go about determining where to put your initial efforts?
Yeah, no, absolutely. And look, that's a terrific question, right? Because I think the risk with a platform like this, right, is you can just go too broad and start experimenting with lots of things and never get anything done, right? So first and foremost, I think the beauty of Omega is that we have a team that is operationally very, very solid, right? I think first of all, big asset is your people, are your people and your team, and we have exceptional people who are operationally very strong. They're not just sort of tinkerers and scientists, who are sort of just experimenting. They know how to take programs forward. So that's one piece, right? But now, having said that, right, as we started initially, the company was formed in 2017. I joined early 2019. I've been here there close to 5 years. My chief scientific officer joined right after me. I did, right? And as we were looking at the different aspects of the, exploring the power of the platform, we started figuring out, and we had, let's say, give or take, 20 to 40 different things that we were looking at in terms of genes and diseases, etc. And then as we kept working, some of them took the lead, right? So what we did was whatever took the lead. And then at one point we decided to just take a handful of them and say, look, let's actually create a pipeline which actually reflects a pipeline even today, right? Because what we were doing as a young company with a completely new platform that nobody has worked on before is exploring the breadth and depth of the platform, really pressure testing the corners of the platform. So what we did was we divided the disease world into sort of four areas, right? One was oncology, because like I said earlier, um, epigenetics is a poster child of oncology. So it makes sense to actually think of oncology if you're using an epigenetic approach. In oncology, what you're doing is, controlling uncontrolled cell growth, right? Other end of the spectrum is regenerative medicine, where you're engendering cell growth. Remember, I was talking about the liver example earlier, right? So regenerative medicine obviously became another area to look into. And then monogenic diseases, which are uh, single gene diseases, which is the mainstay of all genomic medicines, is something we had to demonstrate. And then we looked at multigenic diseases, where if you have multiple genes in the IGD, you could control them with a single therapeutic and actually have tremendous impact. So that's how we set up our pipeline. And as we started prosecuting them sort of in parallel, some automatically took the lead, right? And as things started taking the lead, we interrogated that for market fit, unmet patient need, and where we'll have impact. And that's how we then finally defined our pipeline, right? So what we have done is, since then, we've started prosecuting across our pipeline, and our oncology program and CMYC for hepatocellular carcinoma took the lead. And CMYC by itself is an interesting gene because it is considered the holy grail gene in oncology. It has been unsuccessful. People have tried to drug it over the last 40 years with conventional approaches. They have not been able to do that, right? We believe this is the way to do it. So that program automatically took the lead. Another program which is superb is HNF4 alpha, where we are looking at regenerating liver function. That is applicable from everything from NASH to potentially liver cirrhosis. Then we are going after inflammation and immunology with a multigenic IgD, CXCL1, 2, 3, and IL-8. These are four chemokines, and so on and so forth. So that's really how we have prioritized the pipeline. And for future, what we are going to do is, obviously, we added something new into our pipeline now, the deal with Novo, where we are going to look at a target that's unnamed as of now, but looking at obesity management. And as we do more and more partnerships, that's how we will expand the pipeline. Some things we will do internally, and some things we will extensively partner, because there is no earthly way for Omega to actually exploit the entire breadth of this platform and its capabilities alone.
So obviously the obesity area became very hot last year with the approval of the GLP-1 therapies. What does the Novo Nordisk deal do for Omega? And can you give us some broad brush outlines of how it's structured?
Yeah. So, I mean, look, I think, there are two aspects. One is what the deal does and what it means, and then what exactly uh, is the thing that we are working on, and why us, right? Why did a giant like Novo choose us? So let me actually explain the second part first, right? So if you think about GLP-1s, these are tremendous drugs, right? And they were always being developed for diabetes and obesity. If you talk to people like Uli Stills from Novo, that's always what they have been working on, right? However, the way the GLP-1s work is, for a lack of a better term, they trick us into believing that we are full and we are not hungry, as a result of which we lose weight. There's also another process of gastric emptying. You've heard of people feeling satiated, people feeling nauseous, all of that. That leads to us not eating food. And if you don't eat food, yes, you will lose weight, right? And you have to take these, medicines very regularly based on, whether it's a weekly dosing or whatnot, right? And the downside of this is that you have to take that chronically, because once you stop taking it, your weight returns, and returns with a vengeance, because essentially now you start feeling hungry, right? So while these drugs work beautifully for people, and I think that they're great drugs, because I think it does take care of obesity for people who have no other option, right? There's a downside to it. Now, what if there was a way where you could actually leverage fundamental science to create a much more durable and sustainable uh, approach to obesity? And actually there exists science like that. It's called thermogenesis. So simply put, right, we have two kinds of fat in our body: white fat and brown fat. When babies are born, babies pretty much have almost exclusively brown fat. And brown fat is metabolically active. That fat burns itself off. That's why babies are chubby, but over time they lose all their baby weight. And as we grow up, right? But as we grow up, white fat gets accumulated, and white fat is metabolically inert. It's not active, so it doesn't burn itself off. So simply put, if there was a way to use, you know, epigenetics and gene control to convert or transdifferentiate that white fat into brown fat, which then automatically starts burning itself, right? And doesn't revert back to white fat, you would actually have a much more sustainable way of getting to weight loss. That is precisely the science behind our deal. And as Novo looked at this, and we all aligned on the goals and what exactly we wanted to do. And once they did that, they realized that, well, Omega's platform technology is the one that will actually allow them to do that. Right? That's why we did this deal where we are really working on thermogenesis and converting white fat into brown fat. If this works, this is a complete game changer. There is no reason for me to believe that this won't work. The biology is well known. Our technology is fungible across every disease biology. So I have complete confidence that we'll be able to modulate the genes. Obviously, we'll have to prove it in the clinic, and that will take some time. But that's really the excitement of this. That's sort of the science of what we are doing in this thermogenesis deal. But the other question was, what does this mean for Omega? And that is actually extremely important, right? This is a huge validation for Omega's platform and technology. If you step back, right, when you are on a pioneering journey like Omega is on, where you're creating a completely new class of drugs that nobody has seen before, the understanding of a platform has been there. People get it. If you talk to people in big pharma, they get it. But typically they would like to see clinical data. They would like to see it proven out, right, before anybody starts putting a bet. I mean, go back to sort of the 1970s. I imagine this happened with monoclonal antibodies when Roche first Genentech actually introduced monoclonal antibodies for the first time. And then everybody started working on monoclonal antibodies, right? Now, there are people who are working on what we are doing. They're several years behind. But for us, being on a pioneering journey, it's a huge validation that this platform works, and which we have, by the way, proven in the clinic, right? We have clinical data. We have delivered proof of platform in the clinic. This validation allows a giant like Novo, which is well known in research and in the field of obesity, to say that, look, for the next generational obesity drug after GLP-1s, we want to really work with Omega to tackle some of this fundamental science of thermogenesis, because we believe that that is the platform that can help. That is a huge vote of confidence and validation, and we feel that that's tremendous recognition for Omega.
How far will Omega take a candidate, and is there some decision point for Novo?
Yeah, so the way this deal is structured is that we'll be working, on what we are very good at, which is obviously, doing the early part of the work, discovery, coming up with the epigenomic controllers, right? And then working on a few targets. And then what will happen is, once we get to a development candidate stage, Novo will take a license, and then Novo will actually start doing the work and taking it into IND and beyond. Because Novo really, at the end of the day, are the experts in obesity. Omega is not a company that is going to develop internal expertise in obesity beyond what we need at this stage, right? So that's the deal. It's a real partnership. They will be involved, obviously, in the science, etc., right from the get-go. We'll be involved with them even after we hand it over to them, to make sure that as they take it through IND, we understand our drug better, our the epigenomic controller better. We have already taken it through IND. So it's a real partnership, but that's where the handoff will happen, and they will take a license, and then they'll take it to IND and beyond.
Given the broad potential of the platform, how are you thinking about the pursuit of other partnerships?
Yeah, I'm hoping, Danny, that this actually precipitates many more partnerships, frankly, because we have been talking to big pharma partners for a while. People understand this, but I think, like I said, with a completely de novo technology, sometimes it takes time for people to really wrap their heads around and start believing that this is not, a research project, right? And it's not. We are in the clinic. We have demonstrated that this platform works, preclinically in several disease areas. Now we have proved it in the clinic. We have a Novo deal. So I'm really hoping that this starts precipitating many more partnerships. And, that's the only way. And the reason this is important, by the way, is not just for Omega to have partnership. The reason this is important is because this platform is so broad and it is applicable to so many different diseases that as an ecosystem of biotech and big pharma and investors, if we are not able to exploit this platform, we are probably putting on the back burner some of these path-breaking drugs that could help patients that have no other treatments available to them. Right? Think about c-Myc. Nothing has worked for the last 40 years. People have tried to drug that gene unsuccessfully for 40 years. If our platform works, which there is every indication that it should, because how we approach it, that we have overcome all of the earlier issues with targeting c-Myc. So that tells me that this should work eventually in the clinic. Obviously, we have to prove it in the clinic. But this is the only way potentially to create a c-Myc drug that would work. We don't want to leave that on the table. That's why partnerships are important, because we can't do this alone.
Omega went public in 2021. It's been a rough few years for public biotechs. The stock is down from its $17 IPO and trading below $5. It did bounce on the Novo deal, but what's the discussion with shareholders like these days?
Look, I mean, honestly, I think everybody understands the markets, right? I mean, I think, 2021, 2022, 2023, now we are in 2024, right? I mean, the markets have been, to put it very mildly, it's been a wild ride. Look, at the end of the day, right, what is most important for Omega and a team, my team, and, a company like us and our shareholders, and is that we keep the eye on the ball, right? We are well capitalized. We have money, right? Look, stock price is one way to look at it, but honestly, at the end of the day, we control the controllables, which is everything that is in our control. If you look at Omega, every commitment that we have made publicly to our investors, to our shareholders, right from doing a crossover round C, which was in the early 21, IPO and beyond, we have kept our promises and we have delivered on every promise made. Operationally, this is a very, very strong company. To take a completely de novo platform like our platform, when the company was started in 2017, to get an IND cleared in 2022 is incredible. In a completely new way of even thinking about medicine. That's what our shareholders understand. So we have some really great investors. They're very sticky, long-term investors. Right? We control the controllables. I can't control the stock market and the stock price. So what we do is we keep our eye on the ball and we continue to deliver on our commitments and on our operational priorities. That's the best that I can do. And then look, at some point, if we could come out of the dot-com bust of the 2000 and the 2008 stock market crash, I think we'll be coming out of this as well.
Well, how far will existing cash take you, and what's the plan for raising additional capital?
Yeah, look, I mean, what we have publicly said is that at the end of last year, our last disclosure was the third quarter, that we have close to 90 million in cash, and that will carry us, into third quarter of 2024, right? That's what we have publicly declared. Look, I mean, I think there are many options available, right, in terms of raising money, doing BD deals, etc. And we are always considering all of those. And those options remain available to us. Our shareholders, investors are all extremely supportive. So more on that when we do something. But look, I think we feel very fortunate to have a set of investors that believe in the company, and we use our capital very wisely. So I'm not very concerned about that. I think the company has tremendous longevity.
Mahesh Karande, President and CEO of Omega Therapeutics. Mahesh, thanks so much for your time today.
Danny, thank you very much. Thanks for taking the time to let me talk about Omega Therapeutics. It's a super exciting company.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.